Hi, and thanks for watching. This is part one of the urinary system. And in part one, I'll cover an overview of how the kidneys contribute to homeostasis. And we'll spend most of our time talking about the methods by which the kidneys produce urine, the basic methods. And in part two, we'll add to that by talking about how the kidneys are dynamic, how they're able to adjust their function to meet changing homeostatic demands. So in part one, I'll give you an introduction to the basic anatomy of the urinary system, and we'll talk about an overview of urinary system function. And then we'll launch into the basic mechanism by which the kidneys filter blood and produce urine. We'll apply those concepts to a disorder known as glucosuria. We'll talk about how the kidneys they play a role in maintaining the pH of your blood. And then we'll talk about the rate at which kidneys are able to remove substances from your blood, and that's called renal clearance. So first, the anatomy. As far as organ systems go, the urinary system is a fairly simple organ system. The urinary system includes two kidneys, a right kidney and a left kidney. The kidneys are the urine-producing structures of the body, and that newly formed urine is drained away from the kidney in a tube called a ureter. There's a right ureter and a left ureter, and both ureters drain urine into the urinary bladder. There's just one urinary bladder, which is a storage sac for urine. And when urine is eliminated from the body, it drains through a single tube called the urethra into the external environment. Here we're looking at the basic anatomy of just one kidney. And if we look at the image in the top left, we can see that a kidney is kidney shaped, like kidney beans are kidney shaped, where there's a pinched in region called the renal hilum. And it's at the hilum where three tubes connect with the kidney. In red, we see the renal artery. In blue, we see the renal vein. And in white, we see the ureter. The renal artery delivers oxygen-rich and also waste-rich blood into the kidney. The renal vein drains oxygen-poor blood that has recently been filtered, so it has a lower waste content. The renal vein drains that out of the kidney, and the ureter drains newly formed urine, and that urine is headed for the urinary bladder. In the larger image on the right, we see a frontal section of a kidney. And the kidney consists of three major regions. In light tan, so I'm sort of highlighting it here, in light tan, that's the region known as the renal cortex, the superficial or the outermost region. And then the renal medulla is made up of these tan triangular shaped structures. Collectively, they're known as the renal medulla. And the white structure here is known as the renal pelvis. The urine-producing structure of the kidney is called a nephron. And there is one nephron shown here, and it's this structure right here is a nephron. The nephron is a microscopic structure, and we can see that the nephron resides partially in the cortex and partially in the medulla, and we'll take a more detailed look at that in a little bit. The nephron creates urine, and that newly formed urine drains from the nephron right here at a structure called the renal papilla, and it passes through a minor calyx into the major calyx, and the major calyces all drain into the renal pelvis, and from the renal pelvis, the newly formed urine passes into the ureter, and it's headed toward the urinary bladder. So as we do with any new organ system in this class, we start with big, broad brushstrokes, and I present an overview of how this organ system contributes to homeostasis. And so these are the two basic ways in which the kidneys contribute to homeostasis. Number one, your kidneys are responsible for modifying, for regulating many of the characteristics of your blood. What is your blood made out of? How much of each component is present? And then your kidneys secrete a few things into the blood. And so let me spend a little bit more time on each of these. The kidneys regulate many characteristics of blood. We have not yet talked about the composition of blood, so let me introduce you to that. Blood is a liquid tissue. It consists of a variety of blood cells that are surrounded by or suspended in a liquid called plasma. And so if we take a sample of whole blood out of the body and we put it in a centrifuge, we can separate the liquid component from the cellular component. And we can see that blood is normally 55% liquid, and that liquid is called plasma. And the other 45% of blood is made up of a variety of cell types. And we'll talk more about those when we get to the cardiovascular system. The urinary system primarily deals with modifying the composition of blood plasma. So let's dive in more deeply there. Like any body fluid, blood plasma is mostly water. And the most abundant type of dissolved solute in plasma is protein. And within those proteins, we have albumins, globulins, fibrinogen, and others. So far, we've talked a little bit about chaperone proteins. If you remember, chaperone proteins chaperone or accompany lipid-soluble signaling molecules when they travel in blood. And so some of the albumins are chaperone proteins. Globulins are immune system proteins. Fibrinogen is a protein that is involved in the formation of a blood clot to help you stop bleeding. And then other solutes include electrolytes. We've been talking about electrolytes quite a bit. Electrolytes are charged particles that give rise to physiological electric signals. And so we've been talking about sodium, potassium, chloride, calcium, and others. Nutrients include things like amino acids and glucose. And glucose, for example, that supports ATP production in all of our body cells. Gases include oxygen and carbon dioxide. Regulatory substances mean enzymes and hormones. And waste products. We'll talk a lot about waste products in this chapter. All right, so your kidneys filter blood and take waste products and incorporate them into urine. So your kidneys modify the composition of your blood, and to do that, they require a hefty blood flow. So your kidneys receive between 20 and 25% of your resting cardiac output. What's that? We haven't covered the cardiovascular system yet. So resting means that your body's not really engaged in physical activity. You're not exercising. You're not walking around. And so most of us are sitting here watching this lecture, and yeah, we're at rest. Some of you might be exercising while you're watching this lecture. Maybe you're on the elliptical machine. Good for you. But the rest of us are pretty much resting. And so cardiac output is the volume of blood that your heart ejects in 1 minute. So over the course of 60 seconds, 20 to 25% of the blood that your heart pumps goes to your kidneys. So it gets about a fifth of your blood. And in this slide, we see some of the vasculature of the kidney. We are not going to study the anatomy of the blood vessels that deliver blood to all parts of the kidney. I do wish for you to know the renal artery, of course, the renal artery and the renal vein. Notice here that the renal artery quickly branches into smaller arteries followed by arterioles that deliver blood to all regions of the kidney. And what we are most interested in beyond the renal artery is at the microscopic level, which is not really shown here. So we will be interested in the blood supply to the nephron, which comes at the ends of these fine branches that you can see in the renal cortex. The kidneys regulate many of the characteristics of blood, meaning the composition of blood. And in order to do this, the kidneys need to be able to remove certain substances from the blood and to leave other substances in the blood. And your kidneys determine which substances leave the blood and which substances remain in the blood. And we'll talk a lot about how that's done. So substances that are removed from the blood are substances that are either waste products or substances that are too abundant. There's too much of them in the blood. And so what we see here, these are all waste products: ammonia, bilirubin, uric acid, creatinine, and it's definitely not a comprehensive list. These waste products all arise from normal cellular metabolism, and all cells give rise to wastes. And as multicellular creatures, we need to have a way of eliminating that cellular waste from the body, and your kidneys and your respiratory system are the primary mechanisms for eliminating those waste products from the body. The example wastes shown here I chose specifically because they come up again for us down the road in future chapters. On the left, we see waste products ammonia and urea. And what's shown in that image is a liver, blood vessels, and the kidney. So whenever we use an amino acid for a metabolic purpose, either we're using the amino acid to build new protein, or we're using the amino acid as a fuel source for generating new ATP, the very first step is called deamination. When we deaminate an amino acid, we strip the amino group from that amino acid, and that amino group becomes ammonia, shown here as NH3. And ammonia is toxic. It can't be allowed to build up inside or outside our cells because it can very quickly cause dangerous pH changes. And there's a pH gradient that occurs inside our mitochondria that is very important for oxidative phosphorylation. And so we'll talk about that down the road when we get to the metabolism chapter. So ammonia can be removed from your blood by your kidneys. But more importantly, that ammonia is not allowed to exist for very long. It's very quickly converted into urea. Urea is a non-toxic, very water-soluble substance. It's a very safe substance, but it's still a waste product, and it needs to not accumulate in our blood. And so our kidneys can readily remove urea from our blood and incorporate that urea into urine. In the panel on the right, we're looking at hemoglobin recycling, which we have not talked about yet, but we will. So long story short, your red blood cells have a finite lifespan. They're only around for a few months, and then they wear out and they need to be recycled for parts. And some of that hemoglobin molecule is very valuable and gets reused. And other parts of that hemoglobin molecule are wastes. They're not valuable and they need to be eliminated from the body. And the waste product specifically that we're going to deal with here in the urinary system is bilirubin. Your kidneys also eliminate hormones and other metabolites, other products of metabolism. And I have a few examples shown here. On the left, we see creatinine. Creatinine is a breakdown product from creatine. And we talked about creatine and creatine phosphate that occur inside skeletal muscle cells. Well, it's always being degraded into creatinine, and that creatinine ends up in your blood, and it needs to be eliminated. It can't be allowed to accumulate. And then on the right up top, we see a home pregnancy test. And a home pregnancy test is a urine test. And what that reagent strip is testing for is the presence or absence of a particular signaling molecule. It's a hormone called HCG, or human chorionic gonadotropin. And this signaling molecule is produced by the fetal cells. So the mom's body does not make the signaling molecule, but the fetal cells do. And that signaling molecule, that hormone, gets into mom's blood. That hormone gets cleared from her blood by her kidneys. That hormone ends up in urine. And the only time that hormone is in your blood or your urine is in or around pregnancy. So it's a pretty reliable indicator that there's a pregnancy involved. And then down below, we see our cute little asparagus stalk. And in class, here's where I ask the group, raise your hand if you have experienced asparagus pea, and many hands go up. And then I say, raise your hand if you've eaten asparagus, but you have never experienced asparagus pea. And a few hands go up. And here's where I explain that we all make asparagus pea. When we eat asparagus, the plant body includes a phytochemical that gets metabolized. It gets broken down into metabolites, breakdown products. And one of these metabolites has a particular smell. It's not a good smell. It's a pretty foul smell. And that's what gives rise to the bad odor of asparagus pea. And so if you eat asparagus, you will make this metabolite and your urine will stink. For those of us who have eaten asparagus, but we don't know what asparagus pea smells like, it's because we don't have the olfactory receptor to detect that odorant molecule. And it's genetically determined. So we all make asparagus pea, but not all of us have the ability to smell asparagus pea. And here's where I tell you about the stupidest extra credit assignment that I've ever given. It was for a non-majors class many years ago, and I told the students they could earn a couple more points on their previous exam if, for the upcoming urinalysis lab activity, if when they came to class, if they could produce a urine specimen that was either beet pea or asparagus pea, they'd get a couple of extra points, extra credit. Well, the beet pea is fine. That's a visual assay. That's the pink tinge that urine takes on after you've eaten enough red beets. But the asparagus pea, that's not a visual assay. So I ended up having to smell about 40 people's urine, and that wasn't fun for me at all. In the process, I learned that there's also coffee pea and onion pea. So this is why I don't give extra credit anymore. Continuing to think about substances that your kidneys either leave in the blood or remove from blood as part of manufacturing urine, we can think about ions. So all of these ions, sodium, potassium, calcium, etc., all of those ions are present in blood plasma, but in the right quantity. And so your kidneys can help maintain the proper chemical composition of blood by regulating its ionic composition. Your kidneys contribute to the pH of your blood by modifying how much hydrogen ion or hydroxide ion is present. We'll talk more about that down the road. Your kidneys contribute to determining the overall concentration of your blood plasma, your blood osmolarity, by influencing how much water versus solutes are present in blood plasma. And your kidneys contribute to determining the volume of blood that's in your body, how much blood is in your body right now. And your kidneys can modify that by either retaining water or helping your body to eliminate water. And when your kidneys eliminate more water, your blood volume goes down. And there's a relationship between blood volume and blood pressure. As your blood volume goes down, for example, your blood pressure will also go down. And so the substances shown here, water, acids, sodium, those are all always present in your blood, but they need to be present in the right ratios. And the amount of these things that you take into your body on a daily basis differs quite a bit. On some days you drink a lot of water, on some days you drink very little water. On some days you eat a lot of foods that might have a low pH, on other days not so much. Some days you might eat really salty foods, other days not so much. And so for all of these substances, when they're present to excess, your kidneys are able to reduce the amounts by removing that substance from the blood, and it gets eliminated from the body in urine. And when these substances are present in just the right amount, your kidneys are able to maintain that by leaving those substances there in the blood, by not removing them. Here we see the normal composition and characteristics of urine. On the left, we see that urine is mostly water, just like any body fluid. And we see that the most abundant solute in urine is urea, and that, you'll recall, is a waste product derived from protein metabolism. On the right, we see some additional characteristics of urine. It has quite a flexible pH range. Between 4.5 and 8 is considered normal, and that's an incredibly wide range. If you consider that the normal pH range for blood plasma, is 7.35 to 7.45. So that's a narrow range, but your urine is quite flexible, 4.5 to 8. The osmolarity of your urine is also quite flexible. If we consider that blood plasma has an osmolarity of 300 milliosmoles per liter, and that's pretty vigorously defended, The osmolarity of your urine can range from 65, that's quite a dilute urine, to 1200 milliosmoles per liter. So your most concentrated urine, your darkest urine, can have 4 times the osmolarity of your blood plasma. And we'll talk more about how that happens down the road. Normal urine is sterile, so there are no microorganisms in urine. However, on urine's way out of the body, as that urine passes through the urethra and the external genitalia, it often picks up bacteria, but normal urine is sterile. All right, we're still in the overview of kidney function. In addition to regulating the composition of blood, your kidneys secrete several things into our blood. 1 enzyme, 2 hormones, and glucose. The 1 enzyme that's secreted by our kidneys into our blood is called renin. And renin is the first step in a much larger process known as the renin-angiotensin-aldosterone system. And we will cover that later in part 2. This system is responsible for a whole suite of homeostatic responses that help raise blood pressure, typically in the face of dehydration. We'll talk more about that later. 2 hormones. Calcitriol is the active form of vitamin D. So we may manufacture vitamin D in our skin. We may eat vitamin D in vitamin D rich foods like eggs and certain fishes. But it's not activated until it passes through the kidneys. And the kidneys secrete that activated form, calcitriol. It is a hormone, and it plays a role in blood calcium homeostasis. So for example, calcitriol acts on the cells lining our small intestine, and in response to calcitriol, those cells become more permeable to calcium. So those small intestine cells can absorb more calcium from the food that we've eaten. So calcitriol plays a role in the body obtaining more calcium from the food that it's eaten. And then erythropoietin, or EPO, we have talked about recently in the context of blood doping. EPO is secreted by the kidneys in response to hypoxia. Hypoxia means there's inadequate oxygen delivery. And so certain cells in the kidney can detect that. And in response to inadequate oxygen delivery, these cells secrete erythropoietin. And erythropoietin is this signaling molecule that acts primarily on red bone marrow. And in response to EPO, red bone marrow produces more red blood cells. So the body's response to EPO is to have a higher hematocrit, to have more red blood cells to increase the oxygen carrying capacity of blood. And then our kidneys can also secrete glucose into our blood. We've talked about this just a little bit, but more in terms of the liver secreting glucose into our blood. And so when the body is fasting, if you haven't eaten today, and it's say mid-afternoon and you haven't eaten today, there is glucose in your blood, and that glucose came not from your last meal, but it came from your liver. Your liver broke down glycogen in order to secrete glucose in your blood. We need glucose in the blood to keep living. Your neurons run on glucose. And so in the fasted state, that glucose in your blood is maintained by the breakdown of glycogen in your liver. Under more stressful terms, like a starvation situation, your liver and your kidneys can manufacture new molecules of glucose and secrete those into the blood. So your kidney, as well as your liver, perform a process known as gluconeogenesis. We'll talk a lot about it in the metabolism chapter. And all that word means is the formation of new glucose molecules from non-carbohydrate sources. So if you are on an extended fast or in the starving condition, some of that blood glucose can be manufactured in your kidney. This is a table summarizing what I just went through with you. This table comes from your textbook. Okay, so that concludes our overview of the function of the kidneys. And we will spend the rest of our time here in part 1 talking about the basics of urine formation. How do the kidneys filter blood and take waste products and incorporate them into urine? So during the formation of urine, some substances, particularly waste substances, are removed from the blood and incorporated into urine. And that's how they get out of the body. That's our body's excretion mechanism. To eliminate wastes is to excrete. Other substances need to stay in the blood. They belong there. We don't need to eliminate them from the body. And the kidneys allow them to stay in the blood. There's a third category. Some substances in the blood are removed from the blood by the kidney, but then they're added back into the blood. And so they end up staying in the body and they're not eliminated as urine. So for every component of our blood, every component falls into one of these boxes. It's either removed from the blood and incorporated into urine, or it's never removed from the blood, it just stays there, and so it's never in urine. Or it's removed from the blood and then put back into the blood, and it's not in urine. In order to understand how the kidneys produce urine, we need to have quite a detailed understanding of the nephron. The panel in the right shows one nephron, and that one nephron is a microscopic structure. Each kidney includes approximately 1 million nephrons, and you have 2 kidneys. So we'll be studying the anatomy and physiology of a single nephron, but please don't lose sight of the fact that that one nephron really represents roughly 2 million nephrons. The nephron is composed of 2 different sets of tubes. It's microscopic. The structures that are in gray are collectively known as renal tubules. A tubule is a little tube, a microscopic tube. And the renal tubules have different sections, different substructures. I'll show you that in a minute. Our second set of tubes are blood vessels. And the blood vessels are shown here in red and blue. And let's start with blood flow, because we have these convenient black arrows that indicate the pathway, that blood flows. So we'll begin with a branch of the renal artery. Here, blood flows through this branch, an offshoot of the renal artery, and into an afferent arteriole. The afferent arteriole delivers blood into a capillary ball. This capillary ball, shown here, is not labeled, but it's called a glomerulus. The glomerulus is a wad of capillaries that occur as part of the nephron, and capillaries in general, all capillaries, are leaky. There are literally holes in the walls of capillaries. So as blood passes through capillaries, substances can cross the capillary wall. They can exit the blood vessel or they can enter the blood vessel. Well, here in the nephron, blood pressure, the pressure of the blood against the wall of that capillary, is sufficiently high that it forces water and solutes out of the blood, out of the glomerulus, and into the glomerular capsule. The glomerular capsule is shown there in gray. It surrounds the glomerulus, and the glomerular capsule receives this water and solute that's leaving the blood and entering the renal tubules. The glomerular capsule is the beginning of the renal tubules. And that fluid is called filtrate, which we'll come back to in a moment. If we continue to examine the flow of blood, after blood leaves the glomerulus, it enters a vessel known as the efferent arteriole. The efferent arteriole turns and branches to give rise to the peritubular capillaries. Peritubular. Peri means around. So these capillaries form around the renal tubules. and they have a close physical relationship with each other because some substances will leave the blood and enter the renal tubules, and other substances will leave the renal tubules and go back into the blood. Those processes are known as secretion and reabsorption, respectively, and we'll talk about them in just a moment. Moving on to the renal tubules begin with the glomerular capsule, which I mentioned a moment ago, and the liquid that's inside the glomerular capsule is called filtrate. Filtrate is a body fluid. It's mostly water with dissolved solutes in it, small solutes. Solutes that are smaller than a medium-sized protein. So solutes that include glucose, amino acids, gases, ions are all part of filtrate. From the glomerular capsule, that filtrate moves into the proximal convoluted tubule, shown here. From the proximal convoluted tubule, that filtrate travels down the descending limb of the loop of the nephron. It's labeled loop of Henle here, and that is a correct term for it. There's a trend in anatomy to stop naming things after people, and I'll try to follow that trend. So this is the loop of the nephron. The whole hairpin turn is the loop of the nephron. There's a descending limb of the loop of the nephron and an ascending limb of the loop of the nephron. We moved into the distal convoluted tubule. Here it's just labeled distal tubule. And then into what's known as a collecting duct. A collecting duct services multiple nephrons. And so we can see these little connections that are left bare here, but this is where another nephron would connect to this collecting duct. This collecting duct is ultimately going to drain through a renal papilla and into the renal pelvis, into the ureter, and that urine will make its way to the bladder. Here's a different artistic representation of the same thing. And I'd like to draw your attention to the physical relationship between the blood vessels and the renal tubules. Two important physical relationships. Number one, the physical relationship here between the glomerular capillaries and the glomerular capsule. Remember, the capsule is going to catch the filtrate that leaves the glomerular capillaries. And then number two, the physical relationship between the renal tubules and the peritubular capillaries, shown here. So substances are going to move between the blood and the renal tubules. Some substances are going to leave the renal tubule and enter the blood. That process is known as reabsorption. And other substances are going to leave the blood and enter the enter the renal tubule. That process of leaving the blood and moving into the renal tubule, being added to the filtrate, that's a process known as secretion. In this image, we see a different view of the nephron, where we can determine how the nephron is situated in the kidney. Notice that we see the renal cortex as well as the renal medulla. This nephron is situated partially in the cortex and partially in the medulla, and that becomes important for the formation of a concentrated urine, which we'll talk about later. Notice that the glomerulus, the capillary ball, the glomerulus, and the glomerular capsule right here, or Bowman's capsule as it's also called. The glomerulus and the glomerular capsule both occur in the cortex, as does the proximal convoluted tubule. In this nephron, the loop of the nephron dips into the medulla, and then the distal convoluted tubule is back up in the cortex. And we join the collecting duct. The collecting duct descends deep through the medulla to the renal papilla. The renal papilla is where that newly formed urine drains into the minor calyx, which drains into a major calyx into the renal pelvis. Renal tubules, the walls of the renal tubules, are made out of a particular type of epithelial tissue called simple cuboidal epithelial tissue. It's a single layer of cube-shaped cells. And so if we take renal tubules and we slice them transversely in cross-section, we can see the walls are simple cuboidal cells. So this is the wall of one renal tubule. Here's the lumen. L is for lumen. I don't know which renal tubule that is. I can't tell by looking at the histology. It might be a proximal convoluted tubule. It might be a distal convoluted tubule. It might be the loop of the nephron. We certainly can't tell. They're all made out of simple cuboidal epithelial cells. And here we have renal tubules in longitudinal section. So here's one wall of the tube. Here's another wall of the same tube. And then this is the lumen. It's not clear which direction the filtrate is flowing. It's either going this way or it's going in the opposite direction. But we can see that same tissue now in longitudinal section. And so we've just been looking at the microscopic structure of a nephron, and I want to remind you of where that nephron sits in the bigger picture of an entire kidney. This is the collecting duct here, and the collecting duct will deliver newly formed urine into the renal pelvis, which drains into the ureter. This is a much simplified nephron. We've taken out all the twists and the kinks, and we've made it a lot shorter. Both the blood vessels and the renal tubules are represented here, and the artist took care to represent their close physical relationship. And we'll use this schematic nephron. It's not what a real nephron looks like, but it's a schematic, and it's simplified so that we can more easily understand the processes that occur during the formation of urine. These are the three processes that occur in the formation of urine. Urine formation always begins with glomerular filtration, and then filtration is followed by these two processes, reabsorption and secretion, and these two can co-occur. So step one is always glomerular filtration, and then steps 2 and 3 co-occur in no particular order, as we'll see. So back to our schematic, our simplified nephron. During glomerular filtration, filtrate is produced. And the way filtrate is produced, filtrate is derived from blood. So as blood passes from the afferent arteriole through this glomerulus, as blood is flowing in that glomerulus, as we'll see, the pressure is sufficiently high to force some of the liquid component of blood, water and solutes, out of the blood vessel and into the glomerular capsule here. And now that filtrate, the artist has shown it drawn in a light yellow color. The liquid that we find inside renal tubules is called filtrate. And this is the process known as filtration. So in your mind's eye, can you determine which arrows indicate blood flow toward and away from the glomerulus? Can you point out those arrows? Here's blood flow toward the glomerulus. Here's blood flow away from the glomerulus and toward the peritubular capillaries. Which arrow indicates glomerular filtration? In your mind's eye, can you see that arrow? It's this arrow here. This is showing that some of the components of blood leave the blood vessel and enter the renal tubule as filtrate. The next step in the formation of urine is called tubular reabsorption. During reabsorption, useful substances that are present in this filtrate, useful substances are removed from the filtrate and returned to the blood that's passing through that peritubular capillary. So in your mind's eye, can you pick out which arrow indicates tubular reabsorption? It's all of these arrows show the movement of substances. So this is water and solutes are moving out of the filtrate and back into blood. And then during tubular secretion, the opposite occurs. Substances move out of the blood and into the renal tubule. So they become part of filtrate. And then the final product here is urine. Urine is what drains out of the collecting duct toward the renal pelvis. Here we see a different artistic representation of a similarly simplified nephron. So it's been truncated, it's made shorter, and there are no kinks or twists or loops. And the artist has maintained the close physical relationship between the blood vessels and the renal tubules. We see our three processes represented here: glomerular filtration, tubular reabsorption, and tubular secretion. I selected this image to reinforce the idea that reabsorption and secretion don't necessarily occur in order, they can co-occur. So filtration always happens first. That's step one. But once we have the filtrate, substances can move between the blood and the filtrate in opposite directions. When substances leave the filtrate and enter blood, that's called reabsorption. When substances leave the blood and move into the renal tubule, that's called secretion. And at several points along the nephron, those two processes co-occur. At other places along the nephron, only reabsorption occurs. A metaphor for the movement of filtrate through the renal tubules is the movement of waste in this municipal waste stream along this conveyor belt. The conveyor belt shown here includes genuine waste products, items for which there is no known use, no known value, items that genuinely belong in the landfill, items that need to be packed up and moved out of town to the landfill. This waste stream also includes useful substances, things that do have value, that can be recycled, remade, reused, substances that we don't want to put in the landfill because that would be wasteful. So the useful substances are reclaimed by these conveyor belt workers. Some of these individuals are in charge of picking off all of the aluminum cans. Other workers are in charge of removing all the glass bottles. Other workers are in charge of removing all the paper, aluminum, glass, and paper. Those things are still valuable. They can be used, and we don't want to put them in the landfill. And so the workers along this conveyor belt are like the cells that make up the walls of our renal tubules. As filtrate passes through the renal tubules, substances are selectively discarded or retained. The substances that we want to eliminate from the body, we make sure those substances are on the conveyor belt, because whatever's on the conveyor belt, whatever's in the renal tubule, is going to become urine, and that urine's going to leave the body. Substances that we want to keep in the body, we remove from the conveyor belt. Once we get it out of the renal tubule, we put it back into the blood, and those substances stay in our body. And so the cells that make up the wall of your renal tubules are like these little Lego worker people. Some of these worker people are helping your body hold on to useful substances. Other worker people are helping to eliminate additional waste products. Let's work with these three panels to determine if each substance, X, Y, and Z, is filtered, reabsorbed, secreted, or excreted. Let's start with substance X on the left. Substance X is filtered. To be filtered means the substance moves out of the blood and into Bowman's space. Bowman's space is capsular space, or Bowman's capsule, or the glomerular capsule. So as blood passes through the glomerular capillary, some of that substance leaves the blood and becomes filtrate. This is what it means to be filtered. So yes, substance X is filtered. Is substance X reabsorbed? During reabsorption, a substance moves out of the glomerular filtrate and into the blood. No, substance X is not reabsorbed. There's no evidence in this diagram of the substance leaving the filtrate and returning to the blood. Is substance X secreted? Yes. During secretion, a substance moves out of the blood and into the renal tubule. A substance leaves the blood to become part of filtrate. And so here we see evidence that substance X is secreted. It's leaving the blood and it's becoming part of filtrate. This is secretion. And we see that substance X is excreted. To be excreted means to be eliminated from the body. And everything that's part of urine is excreted. So we see that substance X is filtered, it is not reabsorbed, it is secreted, and it is excreted. Substance Y. Is substance Y filtered? Yes, substance Y is filtered. Substance Y leaves blood as it passes through the glomerular capillary and enters the filtrate. Is substance Y reabsorbed? Yes, here is evidence of substance Y being reabsorbed. During reabsorption, the substance leaves the renal tubule, it leaves the filtrate and becomes part of blood. Is substance Y secreted? No. There's no evidence of substance Y leaving the blood and joining the filtrate. Is substance Y excreted? Yes. Every component of urine is eliminated from the body and so is excreted. So substance Y is filtered, it is reabsorbed, it is not secreted, and it is excreted. Moving on to substance Z. Is substance Z filtered? Yes. Is substance Z reabsorbed? Yes. Is substance Z secreted? No. Is substance Z excreted? No. Substance Z does not occur in the urine. So some of substance Z is filtered, all of substance Z is reabsorbed. None of it is secreted and none of it is excreted. Now that we've covered the basics of filtration, reabsorption, and secretion, we're ready to look at each process in a little more detail. The reality is we could spend the rest of the semester talking about the details of urine formation. We're not going to do that, but I do want to set you up to be able to understand as much detail as you want or have to down the road in your education. So let's take a closer look at how glomerular filtration happens. We can use this image to visualize additional structures that play a role in filtration. What we're looking at is called a renal corpuscle. That's a new vocabulary word. I don't think I've used it before. And an intact, a whole renal corpuscle is shown up top. A renal corpuscle consists of glomerular capillaries plus the glomerular capsule that surrounds those capillaries. So up top, it's shown intact, and down below, the renal corpuscle has been sectioned. It's been sliced open, and this image includes additional important details for filtration. The first of which consists of these yellow cells that have purple nuclei, as shown here. These cells are called podocytes, and they physically wrap around the exterior surface of the glomerular capillaries. Here is the same image of a renal corpuscle with the labels removed. Let's take a look at a renal corpuscle as seen through a compound light microscope. This is a renal corpuscle seen through a compound light microscope. And all of this material here, this is the glomerular capillary. And these pink cells here, these are actually red blood cells that are passing through glomerular capillaries. This space here is the capsular space. So the filtrate will move out of the blood and into this space during the process known as filtration. Over here, we can see a section through a renal tubule. Later on, we'll talk about what the macula densa is, but this is a portion of the loop of the nephron. So filtration occurs when water and solutes leave the blood and become filtrate. And water and solutes move between fluid compartments. Water and solutes move out of the blood and into the capsular space. So inside the blood vessel is one compartment, inside the capsular space is another compartment. And in order to get from compartment 1 into compartment 2, the filtrate needs to cross what's known as the filtration membrane. And so the filtration membrane is the barrier between blood and filtrate. We can use this diagram to understand the filtration membrane. So what we're looking at here is a portion of a capillary that's part of the glomerulus. So let me show you the lumen of the capillary. Right now I'm drawing in the lumen of the capillary. So blood is flowing through there. And the cells that make up the capillary wall, any capillary, those cells are endothelial cells all over your cardiovascular system. This is not specific to the kidney. Your entire cardiovascular system is lined with endothelial cells. And when we get to a capillary, those endothelial cells have holes in them, literally have holes in them, shown here. Continuing on with the wall of the capillary, we find extracellular material shown here in both purple and blue, extracellular material that forms a partial basement membrane. Here it's labeled lamina propria, that's part of the basement membrane. And now we're moving into territory that's specific to the kidney. Wrapping around these glomerular capillaries, we have these cells known as podocytes. And podocytes have lots of radiating processes. These processes are called pedicels, sometimes they're called foot processes, and they literally wrap around the exterior surface of the blood capillary. And these pedicels, they interdigitate. Let me choose another color. We'll pick blue, and I'll draw the interdigitating, or I'll highlight the interdigitating pedicels. And so we have some pedicels coming from above and some pedicels coming from below, and we have this overlapping, interleaving, interdigitating pattern. So these are our pedicels. Well, in between the pedicels, the gaps between them are called filtration slits. And these filtration slits are slits that allow filtration to happen. They're slits that are small enough to exclude anything bigger than a medium-sized protein. So water can cross the filtration membrane, small solutes like glucose, ions, amino acids, anything that's smaller than a medium-sized protein can pass from the blood into the filtrate. The endothelial cells that make up the wall of the capillary, they have fenestrations, those are the holes. And then the podocytes that wrap around the exterior of the capillaries, they have pedicels that interleave or interdigitate to create filtration slits. And this filtration membrane, again, it is a size exclusion technique. It allows smaller substances to cross and does not permit larger substances to pass through. So the filtration membrane is what keeps cells and medium and large proteins inside the blood. Cells and proteins, medium and large proteins, never become part of filtrate under normal circumstances. So perhaps you're familiar with a colander. It too is a size exclusion device. As we pass water over these blueberries, we want the water and any dirt particles to pass through the walls of the colander, but we don't want the blueberries to pass through. The blueberries stay inside the colander, and the water and the dirt particles are able to be filtered out. I love this image. This image was taken using a scanning electron microscope. And what we're looking at here, mostly what we see are the podocytes and their numerous pedicels. Their pedicels interleave to create these tiny, tiny filtration slits. These are the filtration slits here. A healthy body has to always be producing urine. We'll see in the next chapter that the rate at which urine forms can be sped up or slowed down, but a healthy body always needs to be removing waste products from blood and incorporating them into urine. If you think about our cells in a healthy body, all of our cells are metabolically active. So all of our cells are generating waste products that cannot be permitted to accumulate. And so in order to ensure that urine is constantly being formed, we have a suite of anatomical and physiological forces at work that ensure that filtration is always happening. These are 3 factors that encourage or promote filtration, and we'll take a look at each one of these. Let's start with the extensive surface area of the glomeruli. Remember, you have about 2 million glomeruli, and in each glomerulus, there's a lot of surface area as we have these looping networks of capillary. There's a lot of opportunity for water and solutes to cross that surface, to cross that filtration membrane. And then these capillaries are particularly leaky. So these are 3 different types of capillaries that we see in very specific places in your body. And the major difference between them is how leaky they are. And by leaky, I mean how easily does a substance cross the capillary wall? How easy is it for something to leave the blood by crossing that wall? And so most of the capillaries that are in your body are continuous capillaries, the one on the left. So we've recently been talking about skeletal muscle tissue. If you look at the capillaries that course around skeletal muscle cells, they are continuous capillaries. There are gaps shown here. There are spaces in between neighboring endothelial cells called intercellular clefts, and those are openings, passageways for water and other solutes to cross. When we get into the kidney, we have what's known as a fenestrated capillary. Here are endothelial cells. We still have these intercellular clefts in our fenestrated capillaries, but we also have these dots. All of these little dots are called fenestrations. And so fenestrated capillaries are leakier than continuous capillaries. Just to continue on this slide, it's not really relevant for us now, but there are also sinusoid capillaries. And sinusoid capillaries have an incomplete wall. It's not even an intact wall. They occur in the liver and some other places in the body, and they are incredibly leaky. And we have sinusoid capillaries placed strategically around our bodies because there are some places where proteins need to get into and out of the blood. But that's not in a continuous capillary, and it's not in a fenestrated capillary. So we're going to talk a lot about hydrostatic pressure starting now, but we'll continue the conversation about hydrostatic pressures through the cardiovascular system at least. So whenever we have fluid in a container, that fluid exerts a pressure against the walls of its container. So if you have a water bottle in front of you or a mug of coffee, that's fluid inside a container, and that fluid is exerting a hydrostatic pressure against the wall of the container. Here we have blood inside a container. The blood is passing through a container called the afferent arteriole, and then through a container called the glomerular capillaries, and then through a container we call the efferent arteriole. And of course, all of those containers are continuous with each other. And blood exerts a pressure on all of those walls. And we call that pressure a hydrostatic pressure. If you're familiar with blood pressure, that's what we're talking about. What is the pressure that the blood exerts on the walls of these vessels? We call that hydrostatic pressure. And so the blood pressure inside the glomerulus is particularly high. It's higher than in any other capillary in your body. And the reason for this is that glomerulus is flanked by 2 arterioles. The typical pathway of blood flow is from an arteriole through a capillary bed and into a venule. But this is not our setup here in the nephron. In the nephron, we have blood flow through the afferent arteriole, the glomerular capillaries, and then through the efferent arteriole. Arterioles have smooth muscle in their walls. Arterioles have smooth muscle in their walls, and so they are able to change their diameter. They can vasodilate and get wide. They can vasoconstrict and become more narrow. And the dilation and narrowing of the afferent and efferent arterioles can help maintain high glomerular hydrostatic pressure. The word gradient does not appear on this slide, but that's really what we're getting at. Whenever a substance moves from point A to point B in a living system, and you want to understand why, examine the gradients that are present. Gradients are a common theme throughout physiology. And the gradients that we've thought the most about so far are electrical gradients and chemical gradients, or electrochemical gradients, drive the movement of ions across the membrane. But that's not relevant here. What we're talking about here are pressure gradients, where water and solutes are going to be driven from an area of high pressure to an area of low pressure. And there are three different pressures that influence filtration in the nephron. The first two are hydrostatic pressures. So there's going to be a fluid inside a container, and that fluid will apply pressure to the walls of that container. The first one, glomerular capillary hydrostatic pressure, that's going to be blood pressure inside the glomerular capillary. Pressure number two is the hydrostatic pressure that the filtrate places on the wall of the glomerular capsule. So the pressure inside the capsular space. Those two are hydrostatic pressures. More on those in a little bit. Our third pressure is an osmotic pressure. That's new for us, and I'll use the right slide to explain that when we get there very shortly. But these three different pressures are going to sum. We're going to add them up. Some of them promote filtration, meaning they lead to the movement of water and solutes out of the blood and into the capsular space. Some of these forces inhibit or slow filtration. We're going to add them all up, and as we'll see, it always adds up in such a way that filtration will happen. Filtration never stops. We can use this diagram to think about all three forces, but first I want to tell you specifically about the hydrostatic forces. This diagram is very good for understanding the two hydrostatic forces that influence filtration. And there are two hydrostatic forces represented here because we have two different fluids, blood and filtrate, in two different containers. Blood is inside the blood vessel and filtrate is inside the glomerular capsule, or Bowman space, as the author has indicated. So let's think about blood. Blood is inside this blood vessel, and it's going to exert a force against the blood vessel wall. Here, the author has used the terminology PGC to represent the hydrostatic force of blood against the wall of the glomerular capillary. So PGC might be a shorthand for pressure inside the glomerular capillary. We have our second fluid and container here. This is filtrate inside the glomerular capillary. And this filtrate also exerts a pressure against its walls. And the author of this diagram has used the terminology P sub BS. And that might mean the pressure inside Bowman space. This filtrate exerts a pressure on its walls. So let's take a closer look at this pressure inside the glomerular capillary, the blood pressure. So P sub GC, or glomerular capillary hydrostatic pressure, pressure inside the blood vessel, is particularly high. It's at 60 millimeters of mercury. Millimeters of mercury is a unit to express pressure. Maybe you recall it from an expression of blood pressure. We'll talk more about that when we get to the cardiovascular system. Inside our glomerular capillaries, the blood pressure is especially high. 60 millimeters of mercury inside a capillary is the highest capillary pressure in the body. We'll return to this diagram when we get to the cardiovascular system, but I want to introduce it to you now. Shown on the vertical axis, we have pressure measured in millimeters of mercury, and on the horizontal axis, we have blood vessels in order of moving away from the heart. So blood is ejected from the heart and into a large vessel called the aorta. Aorta branches into many different smaller but more numerous arteries. Arteries branch into smaller and more numerous arterioles. Arterioles give rise to capillaries. Capillaries drain into venules. Venules merge together to form larger veins. And the two largest veins in the body are the superior and inferior vena cavae. So along the horizontal axis here, we have the pathway that blood flows away from the heart, through the capillaries, and then back to the heart. So we can look at the pressure inside the cardiovascular system as blood moves away from the heart, through the capillaries, and back toward the heart. Nothing in this graph is specific to the urinary system. Rather, this graph is specific to the cardiovascular system. What I really want to show you is the pressure that are in the capillaries, in systemic capillaries. So this is not including your kidneys. So as blood moves through a systemic capillary, say capillaries in your bicep brachii, the pressure in that capillary drops from just below 40 millimeters of mercury to about 20 millimeters of mercury. If this diagram showed capillaries in the kidney, glomerular capillaries, the pressure inside glomerular capillaries is way up here at 60 millimeters of mercury. So that's almost twice the pressure that's in a systemic capillary. So the blood hydrostatic pressure in our kidneys is extra high in order to promote filtration. The pressure exerted by the filtrate onto the capsular wall is much lower. It's about 15 millimeters of mercury. And so if we take 60 millimeters of mercury inside the blood vessel and pit that against 15 millimeters of mercury inside the capsular space, the 60 millimeters of mercury is going to exceed the 15 millimeters of mercury, meaning water and solutes will move from an area of higher pressure to an area of lower pressure. And then we have to add in our third pressure. This is not a hydrostatic pressure, but rather it's an osmotic pressure. And this might be the first time that we've encountered an osmotic force or an osmotic pressure. An osmotic pressure is exerted whenever we have some type of solute that's trapped on one side of a semi-permeable membrane. So water can cross the membrane, but this particular solute cannot cross the membrane, and it will attract water molecules toward it. And that attraction is what's known as an osmotic pressure. We can use this diagram to understand osmotic pressure. In this diagram, we have two fluid compartments separated by a semi-permeable membrane. On one side of the membrane, we have pure water. On the other side of the membrane, we have a mixture of water and sugar. The membrane is freely permeable to water, and the membrane is not permeable to sugar. And so water will move by osmosis until it reaches a point of equilibrium. The sugar, however, cannot move. An osmotic force is generated by solutes that are trapped on one side of a semi-permeable membrane. So the sugar molecules trapped here exert an osmotic pressure. They tend to attract water molecules, and this is the driving force behind osmosis. So continuing with this diagram, if we can now apply it to the situation in the kidney, where we have two fluids separated by a semi-permeable membrane. We're going to call this blood, and this blood is inside the glomerular capillary. We're going to call this filtrate, and this filtrate is inside the glomerular capsule, or in Bowman's space. And the membrane that separates blood and filtrate, this is the filtration membrane. The filtration membrane is semipermeable. It's freely permeable to water and smaller solutes like ions, nutrients like gases, like glucose and amino acids, but it's impermeable to medium and large proteins and anything larger than that. So now we can think of those black dots as representing medium and large proteins. Those plasma proteins exert an osmotic pressure because they are trapped on one side of a semi-permeable membrane. Those stuck plasma proteins tend to attract water molecules toward them. And so the osmotic pressure exerted by blood is going to retard or inhibit or slow down the process of filtration. So the osmotic force due to plasma proteins in your textbook, that's abbreviated by the pi symbol sub GC. So osmotic force, sometimes called oncotic pressure, is often represented using the symbol pi. And so it's the osmotic force inside the glomerular capillary, pi sub GC. And that's caused by medium and large proteins that are stuck on one side of the membrane. They're trapped inside that glomerular capillary. And they exert the osmotic force, which opposes filtration. And we can measure that force, and that force is roughly 29 millimeters of mercury. So in order to determine the overall force that causes filtration, we need to sum the three forces that we've been talking about. One force promotes filtration. The other two forces oppose filtration. So here we have net glomerular filtration pressure. So we're going to sum the forces and come up with the net force. This is the pressure inside the glomerular capillary. This is the pressure inside Bowman's space or the capsular space. And this is the osmotic pressure inside the glomerular capillary. I see now that this is a typo. This should be P sub GC, the pressure inside the glomerular capillary. Returning to this diagram, we can look at all three forces. Let's go back up here and examine the arrows. So the fattest arrow, indicating that it's the largest force, is the pressure, the hydrostatic pressure inside the glomerular capillary, and that promotes filtration. The two smaller arrows are both pointing in the opposite direction. And they're pointing in the opposite direction because they inhibit or oppose filtration. Up top, we have the pressure in Bowman's space, and down below, we have the osmotic pressure inside the glomerular capillary. And so down here, we can take typical values expressed in millimeters of mercury. We take the major force that promotes filtration, 60 millimeters of mercury, and we subtract from that 60 the values of the two forces that retard or inhibit filtration, and we come up with a net filtration pressure of 16 millimeters of mercury. That's a positive value, and it indicates that water and solutes are always going to move out of the blood and into Bowman's space. This will become important later on in part 2 when we talk about the mechanisms by which urine formation can be adjusted. One of the ways in which urine formation can be adjusted is by modifying the rate at which filtration occurs. How quickly do water and solutes leave the blood and become filtrate? We can speed that up, we can slow that down, and the way we do that is by altering the balance of these forces. So that's to come. Here we see that same math, and I apologize for that same typo. This should be P sub GC if it stands for the hydrostatic pressure inside the glomerular capillary. and we subtract from that the hydrostatic pressure inside Bowman's space or the capsular space. And from that, we subtract the osmotic pressure inside the glomerular capillary. Here's the next thing to think about. What would happen to net glomerular filtration pressure if, for some reason, there were fewer than normal proteins in the blood, specifically medium to large proteins? If we had not enough of them, how would net glomerular filtration pressure be affected? Would net glomerular filtration pressure increase or decrease? Net glomerular filtration pressure would increase because the osmotic pressure due to plasma proteins would decrease. We're going to talk about the rate at which filtrate forms. How quickly do water and solutes leave the blood and become filtrate? How much filtrate do both your kidneys make in a minute? That's the glomerular filtration rate, the amount of filtrate produced in both of your kidneys in 1 minute. And we express that in milliliters per minute. So it's a rate because it's over time. And that largely depends on body size. And so we see a gender difference here, and that's mostly driven by body size. So let's think about how much fluid we're really talking about. In 1 minute, both of your kidneys are performing filtration. Water and solute is leaving your blood and becoming filtrate. And in 1 minute, we'll just take values for the typical female body. In the course of a minute, you make 105 milliliters of filtrate. And to class, I usually bring in a small graduated cylinder to give students a visual sense of what 105 milliliters looks like. And then in 10 minutes, you make more than a liter of filtrate. And a liter looks like a typical water bottle. That's a liter. Just sitting there for 10 minutes, you make a liter of filtrate. And over the course of a 2-hour lecture, you make more than 12 liters of filtrate. That's 2 to 3 times your entire blood volume. You generate that much filtrate in 2 hours. So in a typical day, we make between 150 and 180 liters of filtrate, so that's over 24 hours. But in a typical day, we only excrete about 1.5 liters of urine. So we make all that filtrate, but only a little bit of urine. Why? For the last few minutes, we've been focused on some of the details around filtration, but don't forget, after filtration, reabsorption and secretion occur, and during reabsorption, 99% of the filtrate is removed from the renal tubules and returned to the blood. So 99% of the water gets returned to your blood. All of your glucose, all of the amino acids, most of the ions are reabsorbed, which is why we excrete such a relatively small amount of urine given the vast quantity of filtrate that we make in a 24-hour period. To further that point, we can use this table to compare the composition of filtrate with the composition of urine. So we can use this table to answer these two questions. Which substances are completely reabsorbed and which substances are partially reabsorbed? So let's start with water. Water, it looks like, is partially reabsorbed. We filter 180 liters per day, but we excrete 1.8 liters per day. So most of that water gets reabsorbed, but not all of it. Sodium is also partially reabsorbed. In our filtrate, we filter 630 grams on a typical day, and we excrete 3.2 grams of sodium. So most of that sodium was reabsorbed, but not all of it. Glucose. Glucose in a normal body or typical body is 100% reabsorbed. It's fully reabsorbed. So 100 grams of glucose gets filtered a day, and normal urine does not contain any glucose. So glucose is fully reabsorbed. Urea. Urea is filtered, it's partially reabsorbed. We get rid of some of our urea in urine, but some of it stays in the blood. Now it's time to add more details to our understanding of the processes of reabsorption and secretion, both of which are indicated in this diagram. So we need to take a closer look at the anatomy of a renal tubule and a peritubular capillary in order to understand how substances move between them. So here we see filtrate inside the lumen of a renal tubule, and we see blood inside the lumen of a peritubular capillary. The wall of the renal tubule is made up of cells, those simple columnar epithelial cells, and I'm outlining one of those epithelial cells here. These epithelial cells, their surfaces have different names because they perform different functions. If something doesn't have a function, we have no reason to give it a name. These cell surfaces function differently to each other during reabsorption and secretion. So let me change colors here to show you the cell surfaces. The cell surface that is in direct contact with the filtrate, so this cell surface here, this is called the apical cell surface or the luminal cell surface because it faces the lumen. This cell surface is in direct contact with the filtrate, so meaning as water and solutes go flowing by as that filtrate is traveling along the renal tubule, it's directly flowing across the luminal or the apical cell surface. And we can contrast that cell surface with the other three shown here. This one, and this one. And collectively, those cell surfaces are known as the basolateral surface. They are not in direct contact with the filtrate. Rather, they are in contact with extracellular fluid. Here, this cell surface is in contact with extracellular fluid. This cell surface is in contact with extracellular fluid. And this cell surface is in contact with extracellular fluid. Notice that between the tubule cells, those cells are connected using tight junctions. And I'll remind you that tight junctions are composed of pairs of plasma membrane proteins that physically stitch together or join the plasma membrane of neighboring cells, and they create a leak-resistant seal. So in order for materials to leave the filtrate and enter the blood, it's very difficult for the material to pass in between neighboring cells. Rather, the material needs to go through the cells, as shown here in this arrow indicating reabsorption and in this arrow indicating secretion. So we could spend the rest of the semester talking about how all the various substances are reabsorbed or secreted. We're not. Instead, I'm going to present to you some basic principles, and we'll use an example or two, and I hope that sets you up to learn it in greater detail should you encounter that in a class somewhere down the line. But we're not going to cover all of the details this semester. So we'll start with beginning principles, reabsorption and secretion. The vast majority of reabsorption occurs in the proximal convoluted tubule. So anytime we're talking about reabsorption, we're probably in the PCT. Water, sodium, glucose, potassium, other ions get reabsorbed in the proximal convoluted tubule. A little bit of reabsorption occurs elsewhere along the renal tubules. And then secretion is more spread out over the length of the nephron. There's not really any one point on the nephron where I can say most secretion happens here, but most reabsorption happens in the proximal convoluted tubule. So the reabsorption of most substances is linked to the reabsorption of sodium. And this involves secondary active transport. We're going to look at a sodium glucose symporter as the example, but there are all sorts of sodium symporters, sodium amino acid symporters, sodium and other ion symporters. And so what we're going to cover regarding reabsorption, I have to be very specific here because we simply can't cover it all. We're going to look at the reabsorption of sodium in the proximal convoluted tubule, and we're going to look at the reabsorption of glucose in the proximal convoluted tubule, and those two things are coupled. We're going to look at the reabsorption of water in the proximal convoluted tubule, and that will bring us to a couple of concepts, transport maximum and glucoseuria. In the proximal convoluted tubule, most of the sodium gets reabsorbed and all of the glucose gets reabsorbed. And sodium and glucose can be reabsorbed by different proteins. We're going to cover one example, and it's called a sodium glucose symporter. We've talked about symporters before in the context of secondary active transport. We've seen this diagram before. So please recall that a symporter is a transmembrane protein that moves two substances in the same direction simultaneously. So shown here are two examples, a sodium glucose symporter and a sodium amino acid symporter. And both of these occur in renal tubules. And contrast a symporter with an antiporter or a countertransporter. An antiporter, as shown on the left, moves two substances in opposite direction. And our examples here are a sodium-calcium antiporter and a sodium-hydrogen antiporter. The molecule that I really want to focus on in this example is the sodium glucose symporter, moves both substances in the same direction. Symporters are a form of secondary active transport. We can use this diagram to refresh our memory about secondary active transport. Remember that secondary active transport relies on primary active transport. Without primary active transport, secondary active transport cannot occur. And in this diagram, we see both secondary active transport and primary active transport. But let's take a step back for a moment and look at an overview of this diagram, and then we'll go into the specific transport mechanisms. So as shown up top here, we're looking at a portion of the proximal convoluted tubule. We see filtrate inside the tubule here. We see portions of 1, 2, 3 tubule cells. These are the cells that make up the wall of the proximal convoluted tubule. These cells have projections known as microvilli. These are hair-like extensions of the plasma membrane to increase the surface area of these cells. So this is the apical cell surface here, but A is for apical. And the surface area of the apical cell surface has been augmented or increased by this undulating brush border. There's more plasma membrane here. Over here, we have extracellular fluid, and so we have the basolateral cell surfaces of these epithelial cells in contact with the extracellular fluid. And then here we have blood inside a peritubular capillary. So the filtrate is flowing through the lumen of the renal tubule, and reabsorption of sodium and glucose is occurring via secondary active transport. Where's the primary active transport, Laura? I thought you said we must have primary active transport in order to have secondary active transport. That's true. Do you see primary active transport happening here? Of course you do. It's our good friend. Remember that old best friend? Our BFF, the sodium-potassium pump? Yeah, well, it's never left your side. So this is a sodium-potassium pump. It's constantly using ATP to drive sodium out of the cell. Not shown here because it's less relevant here, but it's also pulling potassium into the cell. So this sodium-potassium pump is maintaining a concentration gradient for sodium. When it drives sodium out of the cell, it keeps intracellular concentration of sodium very low. That favors the movement of sodium into the cell from the filtrate. So there is a concentration gradient for sodium that's air quote, pulling sodium or driving sodium into the cell across the apical membrane. This is our symporter here, shown in light blue. This symporter will allow sodium to diffuse downhill, down its concentration gradient from an area of high concentration to an area of low concentration. But the only way it will allow sodium to move is if a glucose travels with it. And so this symporter here in light blue moves both sodium and glucose out of the filtrate and into the tubule cell. This is the reabsorption of sodium and glucose. Sodium is ultimately going to be pumped out of the cell by the sodium-potassium pump. Glucose will be transported across the basolateral membrane by a glucose transporter. So this is facilitated diffusion. Glucose will move from an area of high concentration to an area of low concentration and ultimately into the peritubular capillary. I love this diagram. I found it somewhere online, and we see here the proximal convoluted tubule and the surrounding peritubular capillaries. And there are yellow arrows that are part of the filtrate. As that filtrate flows through the proximal convoluted tubule, notice the width of the yellow arrows decreases across the space. The width of the yellow arrow represents the relative concentration of glucose in the filtrate. So as this filtrate proceeds away from the glomerular capsule through the proximal convoluted tubule, glucose is being reabsorbed, it's being reabsorbed, it's being reabsorbed, so that glucose is leaving the filtrate and entering the peritubular capillaries, such that by the time we get to the end of the proximal convoluted tubule, and now we're at the beginning of the loop of the nephron, there is no more glucose remaining in the filtrate. 100% of the glucose has been pulled out of the filtrate and has moved into the blood in the peritubular capillaries. So that's almost all of our coverage of the reabsorption of sodium and glucose. We'll return to that in just a moment. But we're in a position to talk about the reabsorption of water in the proximal convoluted tubule. And in the PCT, water is going to move by osmosis. Water always moves by osmosis. Where the solutes go, water flows. And so if we're moving solutes, like sodium and glucose, if we're moving solutes out of the filtrate and into the blood, guess what the water's doing? The water's following solutes. So as solutes are reabsorbed, water is as well. And we call this obligatory water reabsorption. Water is obligated to follow solutes. Here's a diagram where we can see obligatory water reabsorption. So again, these are tubule cells in the proximal convoluted tubule. And many of these cells are busy reabsorbing solutes like urea, magnesium, calcium, potassium, chloride, not shown here, but certainly sodium and glucose. And then we can see that water follows by osmosis. Water follows solutes. And so we call this obligatory water reabsorption. Water is obliged to follow solutes. We need to reabsorb solutes because we would die very quickly if we let all of the solutes that are in filtrate, if we let them all stay part of filtrate and eventually become urine, we would pee out our solutes very quickly and die. So we need to reabsorb solutes. Water will always follow those solutes. And so it's obligatory water reabsorption. We can't turn it off. Later on, we'll see that we can adjust the rate of obligatory water reabsorption by adjusting the rate of the absorption of certain solutes, but we certainly can't turn it off. During obligatory water reabsorption, water simply follows the solutes as they are reabsorbed. Later on, we'll cover facultative water reabsorption. And maybe you know that word because you've taken microbiology and you've heard about facultative anaerobes. So in biology, we use the term facultative when something can be adjusted according to need or circumstance. Where a facultative anaerobe is a type of microbe that can adjust its metabolism to a particular environmental circumstance. Is oxygen present or not? And so later we'll cover facultative water reabsorption. It's a different mechanism of water reabsorption that we can turn on or off depending on how dehydrated the body is. In continuing our conversation about reabsorption, we need to talk about transport maximum. So transport maximum is the maximum amount of a substance that can be reabsorbed per unit time, and we express it in milligrams per minute. So for example, in milligrams per minute, how much glucose can your kidneys possibly reabsorb? That's their transport maximum. And it's possible to exceed a substance's transport maximum. And so do you see the image there? I always ask my class, raise your hand if you recognize this image. And over the years, the number of hands that go up decreases. And so this is a scene, a very famous scene from an old television show called I Love Lucy. The creator and star is shown there on the right. That's Lucille Ball, a comedic genius and barrier breaker for women in entertainment. And the premise of the show is that Lucy and her best friend Ethel's there on the left, Lucy and Ethel get into all sorts of trouble. And for this scene, they've taken a job in a chocolate factory. And I'd like you to watch this skit. And so notice that there's a conveyor belt there in the scene. So it's going to continue the metaphor of filtrate moving through the renal tubules, being likened to materials moving along a conveyor belt. And so we have workers. We have Lucy and Ethel, and they're workers, and they need to modify the composition or the contents that are on the conveyor belt. So I'd like you to pause this lecture and go to YouTube and do a search for Lucy Chocolate Factory. Every time I post a link, the link breaks eventually because I think it's a violation of copyright. Somebody posts the scene onto YouTube and somebody else takes it down. So go and do a fresh search, watch the scene, laugh out loud, be amazed, and then come back to this lecture. Lucy and Ethel in the Chocolate Factory elucidate the concept of transport maximum. There are simply too many chocolates on the conveyor belt for Lucy and Ethel to capture and process all at once. So transport maximum can be exceeded when there's too much of a substance in the filtrate. The cells that make up the walls of the renal tubules might be working as hard as they possibly can to reabsorb that substance, and there's simply too much of that substance than can possibly be reabsorbed. We've exceeded our transport maximum. And an example of this will be when glucose ends up in the urine, a condition known as glucosuria. So again, glucosuria is when we have sugar in the urine. It doesn't occur under typical normal circumstances in a healthy body. There's no glucose in the urine because 100% of that glucose that wound up in filtrate, it all gets reabsorbed by the time the filtrate reaches the end of the proximal convoluted tubule. There's no more glucose left in the filtrate. However, under some circumstances, we can exceed the transport maximum for glucose. When there's extra glucose in the filtrate, there can be more glucose than could possibly be reabsorbed by the proximal convoluted tubule. And so under normal circumstances, our blood glucose level fluctuates between 70 and 110 milligrams per deciliter. That's fasting blood glucose. But when you have a higher than normal blood glucose level, above 200 milligrams per deciliter, that's going to translate to a higher than normal amount of glucose in your filtrate. And we're going to exceed the transport maximum for glucose. So how do we get a higher than normal blood sugar level? Two common causes. Number one is excessive sugar intake. So you've eaten the whole bag of candy, or you ate the whole tray of brownies. So it doesn't happen often, but it does happen. You eat the entire pint of ice cream. And then diabetes is a very common endocrine disorder where blood glucose can get excessively high because there's a problem in the insulin system. So when we have high blood glucose, we're going to automatically have high filtrate glucose. Remember, glucose is part of filtrate. And so in the panel up top here, when the person has a normal level of glucose in their blood, they have a normal level of glucose in their filtrate. Their proximal convoluted tubules are well equipped to reabsorb all of that glucose, and we don't see glucose in the urine. So there's no glucosuria. In the panel below, we see what happens when the person has a high level of glucose in the blood. High blood glucose leads to high filtrate glucose. In the proximal convoluted tubule, those glucose transporters can be working at their maximal rate, and we still have too much glucose in the filtrate. We have more glucose than could possibly be reabsorbed by the time we get to the end of the proximal convoluted tubule. And there is no more opportunity beyond the PCT. There is no other opportunity for glucose to be reabsorbed. So if it's left in the filtrate at the end of the PCT, it's stuck there, and it will show up in the urine. And so under this circumstance, we have exceeded the transport maximum for glucose, and we do observe glucose in the urine. There is one other condition that leads to glucose in the urine. It's not very common, and it is a genetic disorder with one of the transporters for glucose in the proximal convoluted tubule. It doesn't function very well. And so this person with familial renal glucosuria, they have a limited capacity to reabsorb glucose in their proximal convoluted tubule. So they may have normal blood glucose levels, but they still have glucose in the urine. They are literally peeing away some of their blood sugar. And so we can see that in the bottom panel. In the very bottom panel, the person has a normal blood glucose. They don't have diabetes. They've not eaten an excessive amount of sugar. They have normal blood glucose, and their proximal convoluted tubules are at their transport maximum. Because they have a dysfunctional glucose transporter, their transport maximum is much lower than the person without this disorder. And so we do observe glucose in their urine. But again, that is a genetic disorder, and it's pretty rare. If you have glucosuria, it's probably because you've eaten a lot of sugar or you have uncontrolled diabetes. I've always wanted to follow my niece around on Halloween because she's permitted to eat as much candy as she wants, and I always wanted to test her urine. She's 16 now, so I don't think she'll go for that. I missed my window of opportunity. All right, so glucosuria, the presence of glucose in the urine, can lead to other symptoms, including increased urine output. The person needs to urinate frequently, and they urinate a lot, which leads to dehydration, having insufficient water in the body, and dehydration can lower blood volume, which can lower blood pressure. And so how are those things connected? So if we've exceeded our transport maximum for glucose, and glucose remains in the filtrate beyond the proximal convoluted tubule, it becomes a solute trapped on one side of a semi-permeable membrane. So imagine we're at the top of the loop of the nephron, and there's glucose present in the filtrate. There's no opportunity for that glucose to get out of the filtrate anymore. There are no more glucose transporters anywhere else in the nephron. And so that glucose is going to travel as part of filtrate through the loop of the nephron, through the distal convoluted tubule, through the collecting duct. It's trapped on one side of a semi-permeable membrane, where the semi-permeable membrane is the wall of the tubule. And so you remember what solutes that are trapped on one side of a semi-permeable membrane do. They exert an osmotic pressure. So that glucose that's inside the tubule is going to attract water molecules toward it. So that's going to inhibit or reduce obligatory water reabsorption. More water will stay in the filtrate, meaning more water will end up in the urine along with the glucose. That's going to lead to higher urine output. You're going to urinate a larger volume of fluid. That triggers water loss. You'll become dehydrated. When we're dehydrated, the total volume of blood in our body goes down. And blood is a liquid confined to a set of tubes. When we reduce the volume of a liquid in its container, the pressure that that liquid exerts against the walls of its container goes down. So we become dehydrated, we lower our blood volume, and our blood pressure also drops. So these are some common symptoms of either undiagnosed diabetes, the person doesn't know that they're diabetic, or uncontrolled or under-controlled diabetes. So in both circumstances, the person either doesn't know they have diabetes or they're not properly controlling their diabetes. Their blood sugar can get excessively high. They have high blood sugar that leads to glucoseuria. That causes them to urinate excessively, which dehydrates them. That leads to excessive thirst. They're thirsty all the time. They can't get enough water. And if we test their urine will turn up positive for glucose. We've been talking about reabsorption, and I'll remind you that most substances are reabsorbed in the proximal convoluted tubule. Some reabsorption also occurs in the loop of the nephron and the distal convoluted tubule. And so by the time we reach the late distal convoluted tubule, let me show you, by the time we get around here, 95% of the water and solutes have been reabsorbed. So we can do some additional tweaking with what's left of our nephron. And what I'd like to do next is to talk about secretion. Let me remind you that secretion is essentially the opposite of reabsorption. During tubular secretion, a substance is moved out of the blood and into the filtrate. And we'll just do one example of a substance that is secreted, and that's hydrogen ions. Hydrogen ions can be secreted by the cells that make up the wall of the collecting duct. I hope when you start to think about hydrogen ions, pH comes to mind. I hope you remember that pH is a measure of the concentration of hydrogen ions in a fluid. And we're going to think about the pH of blood a little bit right now. Blood pH homeostasis we'll cover in greater detail when we get to the respiratory system. But for right now, your blood pH is maintained in a relatively narrow range, just 7.35 to 7.45. And the pH of your blood is super duper important because pH helps determine protein shape. And so we do have that narrow range, and your blood pH is defended by several overlapping homeostatic mechanisms that work hard to keep it within that range. When we get to the respiratory system, we'll talk a lot more about blood pH, but we're at the urinary system. So your kidneys can contribute to raising or lowering as needed the pH of blood by retaining or excreting hydrogen ions and hydroxide ions according to need. So when your blood pH begins to fall, meaning your blood pH is becoming too acidic, there are too many hydrogen ions present in your blood plasma, the kidneys can help by secreting hydrogen ions into filtrate. During the tubular secretion of hydrogen ions move out of the blood and into the filtrate. And we have cells in the collecting duct, the late distal convoluted tubule and the collecting duct, they're highlighted here in blue. Those cells have the ability to move hydrogen ions into the filtrate. And this has the function of helping to raise blood pH. So here we're looking at that mechanism. We are looking at filtrate, shown here. We're looking at a portion of 1, 2, 3 tubule cells. Only here these tubule cells make up the wall of the late distal convoluted tubule and the wall of the collecting duct. And these cells have the machinery required to secrete hydrogen ions. What is the mechanism of transport of hydrogen ions across the apical membrane of these cells? That's it, you got it. It's primary active transport. If you've noticed this protein here, This is a proton pump, or a hydrogen ion pump would also be an acceptable name for it. Notice that it uses ATP, and it's going to be driving this hydrogen ion uphill against its concentration gradient from an area of low concentration to an area of high concentration. And in the secretion of hydrogen ion, we can dramatically lower the pH of urine. Normal urine has a lower pH value of 4.5. That's pretty acidic. And so when we get to the respiratory system, we will return to the regulation of blood pH. But for now, please know that of these three systems that all contribute, the respiratory system, buffer systems, and the urinary system, the urinary system is slowest to act. It takes some time to ramp up to the maximal rate of tubular secretion of hydrogen ions. The cell can synthesize additional hydrogen ion pumps, can synthesize those proteins, and can insert them into the apical plasma membrane. However, that takes time. That takes hours and days to achieve maximum secretion rate. Here we return to the normal characteristics of urine, and I've highlighted the normal pH range for urine. It's incredibly broad. Normal urine can have a pH as low as 4.5 and as high as 8, so it can span from acidic to alkaline, and that's all considered normal because the kidneys do contribute to blood pH homeostasis, and plasma has this relatively narrow range, and blood has this quite flexible range. Urine, has this quite flexible range. And the reason why blood plasma, has a very narrow acceptable range is because we have plasma proteins. And pH determines the three-dimensional shape and the function of proteins. Urine is just a means for carting wastes out of the body. Normal urine doesn't contain any protein, and any protein that is in the urine certainly doesn't have any meaningful function. We don't need it to function. It would be totally fine for any protein that's in the urine to denature, and so it is permissible for urine to have this flexible pH range. Let's use this table to recap what we've covered so far, an overview of urine formation. So we have four substances: water, sodium, glucose, and potassium, and we have three columns. The column on the left is the quantity filtered per day, the middle column is the quantity reabsorbed per day, and the column on the right is the quantity excreted per day. Let's go row by row and then think about what's different for potassium. So water, you filter approximately 180 liters of water in a 24-hour period. You reabsorb most of that, and you excrete between 1 and 2 liters per day as urine. Sodium, you filter, here it's 579 grams. You reabsorb most of that sodium. What you didn't reabsorb gets excreted. 4 grams are excreted per day in urine. Glucose, if you filter 162 grams, you will reabsorb all of that. You will reabsorb 162 grams so that no glucose is excreted from your body in urine. Potassium. If you filter 29.6 grams of potassium, you will reabsorb 29.6 grams of potassium. Well, then how did we get 2 grams of potassium in the urine? The math doesn't add up. It turns out that potassium is filtered, reabsorbed, and secreted. It's part of blood potassium homeostasis. All of the potassium that's initially in the filtrate gets reabsorbed. So 29.6 grams are filtered, 29.6 grams are reabsorbed. But later on, potassium can also be secreted in order to adjust the blood potassium level. Our final concept in urinary system part 1 is renal clearance. Renal clearance is the volume of plasma from which a substance can be completely eliminated, removed, or cleared per unit time. And we express renal clearance in milliliters per minute. We can compare these two very different substances in their clearance rates. So glucose versus creatinine. Glucose is a nutrient. It's an important molecule. We need it in the body. It is not a waste. So glucose clearance is 0 milliliters per minute, meaning it is not removed from blood plasma. Glucose is filtered. It's part of filtrate. So it is removed from blood plasma, but it's all put back into blood plasma. It's filtered and 100% reabsorbed. So no glucose is cleared from blood plasma. So the clearance is 0 milliliters per minute. Compare that to creatinine. Creatinine is a waste product. It comes from skeletal muscle cells. It has no known value in the body. We need to get rid of it. Creatinine is filtered. It's part of filtrate. Creatinine is not reabsorbed. And creatinine can be secreted. And so the creatinine clearance rate is between, for a male, 97 to 137 milliliters per minute. We see the different values there for female. The clearance rate for any given substance varies with body size, gender, and age. The bigger point here is to compare a nutrient molecule where the clearance is 0 to a waste product where the clearance is much higher. A creatinine clearance test is a common test for kidney function. And what the creatinine clearance test really estimates is the rate of glomerular filtration. So in a creatinine clearance test, it involves taking a blood sample and a urine sample, and we compare the creatinine level in the blood to the creatinine level in the urine. And we want them to be similar. We want to know that your kidneys are clearing creatinine adequately. And again, creatinine is filtered, it's not really reabsorbed, and it is secreted. And so if we have an abnormal creatinine clearance test, it looks like this. There's way more creatinine in the blood than there is in the urine. We have a low level of creatinine in the urine, and we have accumulating creatinine in the blood because the kidneys are not excreting it fast enough. So that's an abnormal creatinine clearance test. In a normal creatinine clearance test, we have similar levels of creatinine in the blood and creatinine in the urine, indicating that the kidneys are adequately excreting creatinine. We can wrap up this part 1 of the urinary system. In part 1 of the urinary system, I gave you an overview of the basic functions of the kidney, and we talked about the basic mechanisms by which urine is formed. And as part of that coverage, we talked about how do we get glucose in the urine and what might it mean, what are the consequences. We also talked just a little bit about how the kidneys can contribute to blood pH homeostasis. And then finally, we ended with the concept of renal clearance. So there is a part 2, and in part 2, we talk more about how the kidneys are responsive to changing homeostatic demands.